Assalamualaikum and good morning to all my fellow panelists. My name is Mikhail Haqimie bin Hazrin and I will be presenting my final year project, which is hydrocarbon degrading enzyme of Alkanivorax borkumensis, a narrative review. First of all, the petroleum consists of polycyclic aromatic hydrocarbon and persistent organic pollutants. Petroleum hydrocarbon pollutants have a significant negative impact on the environment. Nearly 200 bacterial, cyanobacterial, algae, and fungal genera, representing more than 500 species and strains, are included in the most recent list of hydrocarbon degrading bacteria, which was created nearly a century ago. There are three problem statements here. First, conventional hydrocarbon removal methods like physical and chemical treatments are costly, effective, and may create secondary contaminants. One of the main sources of petroleum hydrocarbon pollutants released into the environment is the crude oil sector. A. borkumensis plays a critical role in the breakdown of hydrocarbon. It is less known about the precise enzyme it uses, how they work, and whether they could be used in bioremediation. There are four objectives here, which is to investigate the key enzyme produced by marine bacteria. Second, to study the predominant roles of enzyme in hydrocarbon degradation. Third, to illustrate how the enzymatic mechanism of marine bacteria contributes to their efficiency in bioremediation, particularly in the oil-polluted environment. And finally, to assess the current and potential application of A. borkumensis enzymes in environmental management and pollution control. There are two mechanisms of hydrocarbon degradation, which is aerobic and aerobic. Under aerobic conditions, bacteria break down aliphatic and aromatic hydrocarbons using oxygen. The compounds go through oxidation steps to form alcohols like aldehydes and acids. These are then converted into acetyl-CoA and enter the TCA cycle to produce energy. In anaerobic degradation, oxygen is not needed. Alkanes are activated through the fumarate addition pathway, while aromatic compounds follow peripheral and central pathway. Eventually, both types are converted into acetyl-CoA and further broken down for energy. For alkene release, the process occurs in two phases, which involve two types of alkene hydroxylase, which is LAKG and LAKB. LAKG first receives electrons and become reduced. It then transfers the electrons to ALKB, which use them to oxidize N-alkanes into primary alcohols, with oxygen as a reactant and water as a byproduct. Next, lipase functions as by attacking ester bonds through a nucleophilic attack by the enzyme's active serine site. This breaks the ester bond, which resulting in the formation of a carboxylic acid and an alcohol. Both enzymes play a crucial role in breaking down hydrocarbon into simpler and biodegradable compounds. Next is esterase, which function by breaking ester bond and converting ester into acids and alcohol through a nucleophilic attack mechanism. The other enzyme is oxygenase, which has two types. First is monooxygenase, which insert one oxygen atom into aromatic ring or alkyl chains, and then dioxygenase, which insert two oxygen atoms into aromatic compounds, either activating the ring or breaking it open. Lastly, cytochrome P450, which use a catalytic cycle that includes peroxygenase activity and help form terminal alkene by incorporating oxygen atom. Next, I will explain the efficiency of bioremediation by A. borkumensis. Firstly, this bacterium produces a high amount of lipase, which is stable over a wide pH and temperature range, which helping in oil degradation. Secondly, it expresses P450 enzyme like CYP153A1A3, which catalyze terminal hydroxylation of hydrocarbon. Each genome contains multiple P450s that play a key role in marine environment. Next, aborkumensis SK2 produce biosurfactants and optimize carbon to nitrogen ratios are crucial for this process. Lastly, is biofilm release EPS and help trap and digest oil at the water-oil interface through interfacial tubulation. Now I will explain the current and potential application of aborkumensis. This bacterium plays a key role in oil spill bioremediation by transforming toxic hydrocarbon into less harmful compounds and increasing bioavailability through biosurfactant production. It also shows the ability to degrade synthetic polymers like polyesters, which supporting marine ecosystem sustainability. In industrial setting, aborkumensis enzyme can be mass-produced for applications like wastewater treatment and bioproduct manufacturing. Lastly, it forms symbiotic relationship with marine organisms like sea alligator, which helping bacteria colonize and degrade oil more effectively in contaminated environments. And lastly is the adaptation to diverse environmental conditions where the bacteria are capable of breaking down hydrocarbon in a variety of conditions, including the ocean mesopelagic zones. Furthermore, some more labile surface can support the growth of alcarnivorous species. Then there are a few research gaps from this review. Firstly, the entire spectrum of hydrocarbon degrading enzyme that aborkumensis produce is not well understood. Important enzymatic pathways lack well-defined molecular processes, particularly how these enzymes interact molecularly with hydrocarbon, and the catalytic mechanism of ALKB is still unknown. There are few recommendations for this topic. First is investigate enzyme efficiency under extreme environmental conditions such as temperature, pH, and salinity. Second is apply genetic engineering to enhance specific enzyme activity. Next is test its biodegradation potential in real-world waste or industrial scale system, and finally explore its ability to degrade other plastic types such as PET or polystyrene. In conclusion, aborkumensis play a key role in marine oil biodegradation due to its unique enzymatic system. Alkane degradation, especially C10 to C19, which is carbon 10 to carbon 19, is facilitated by enzymes such as alkane hydroxylase, oxygenase, lipases, and esterases. Its genome encodes various alkane oxidation mechanisms, which allowing flexibility towards a wide range of hydrocarbon substrates. This adaptability makes it valuable for biotechnological application, including plastic waste management and bioremediation. Field tests show that enzyme combination from aborkumensis are effective in reducing petroleum pollutants. Thank you.